Okay, this video is a book review, and the name of the book is called The Quest for a Cure. It's written by Brent Stockwell Phd, and the guy's a genius. He's a chemist from Harvard who is heavily involved in drug research, trying to come up with a new drug. He became pretty famous for discovering a type of cell death called ferroptosis, related to the Fenton reaction of iron overload in combination with that causing lipid peroxidation, destruction of lipids, chain reactions, and all kinds of problems. Anyways, the picture here is a bunch of chimpanzees throwing mud, okay? And it's kind of like the old joke of if you throw enough wet toilet paper to the wall, some of it will stick, or you throw enough mud at a wall, some will stick. And that's basically what drug research is like. And the sophistication of these individuals is pretty much what drug research is like. I'll explain that in a moment, but this picture here gives you a sense of what's really going on. Now, this picture here is all the biochemical pathways known to occur in humans. And you can see Krebs cycle right in the center, the tricarboxylic acid cycle, also called the citric acid cycle. And the point to get from this picture is it is incredibly complex. All these molecules have to interact with each other, and it's so complex that the greatest human genius times a million can't fully understand it. And the reason I show it to you is just to get an idea of how complicated it would be to be able to design this entire system and get it to work, to go from a sperm and an egg, create a full-size adult human, and have everything function, for an average of 70, 80 years, whatever. So it's mind-boggling how complex this is. Okay, and an individual cell, I'm going to get into his book in just a moment and what his research has shown and how that's relevant, but just to give you a sense of an individual cell, it's like a big city. You got incredibly concise storage of information in DNA. The human genome has 3.3 billion, that's 3.3 billion base pairs of DNA. Those are the blueprints for building a human, for running a cell, for making proteins and everything else a cell needs to do. The cellular nucleus is like town hall. Glycolysis is like a coal-burning electrical plant. Mitochondria are like nuclear power plants. They split the atom. Plasma membrane ion pumps are like an electrical battery. The plasma membrane is like a city wall or in the toll booth. The vacuole is like a water tower holding water for the cell. Flagella is a motor. Microtubules are like roads or railroad tracks. Rough endoplasmic reticulum, it's called rough because of the ribosomes on it, is a factory for making proteins. There's all kinds of proteins that do weird things. They're like chaperone proteins that help a protein to fold. Those are like nurse's aides, helping a patient get dressed. Smooth endoplasmic reticulum is for making lipids and steroids. Golgi apparatus is like a post office, making sure that whatever is made, the protein, the lipid, the steroid gets shipped to the correct location, either intracellular or extracellular. The lysosome is a recycling plant. Heat shock proteins are like garbage trucks, like HSP70, sending old proteins for degradation to the lysosome. Ubiquitins that mark proteins for degradation, they're like part of a cleanup crew. The immune system is like a bunch of plumbers fixing problems. It's also a cleanup crew. It's also like security guards. It's also like the defense force, so to speak. Clotting proteins are like engineers plugging up holes and leaks and problems. So anyways, these are, what I'm trying to say here is individual cells have figured out who knows how many millions of years ago how to do all kinds of incredibly complicated things. Okay, and the old joke is if you went to the beach and you saw your name written in the sand, you would know an intelligent, literate person had done that. So how could the human genome with 3.3 billion base pairs have happened by accident and be so incredibly complicated and sophisticated and perfect that it could run all those biochemical cycles? It's mind-boggling, okay? The correct response to this is awe and to be mind-boggling. It's only God could do it. I say this after having studied this, evolutionary biology, human molecular biology for many, many years. Ever since I was 17 years old, I've been studying this stuff. a lot. Okay, I majored in evolutionary biology at Stanford. Okay, so I just tell you that people who say atheism explains stuff and Darwinism, they don't know what they're talking about. They're full of BS. Okay, now this guy right here, Brent Stockwell, by the way, his initials are BS, but he's not a BS artist. This guy's a chemical genius. Like I said, he discovered ferroptosis, a mechanism of cell death. He developed a library of chemicals to help scientists try to find new drugs for people. And modern drug design is like alchemy. You're trying to turn some dirt or some cheap stone or rock or metal into gold. It costs over $1 billion to make a new drug and take it to market. Over $1 billion. Imagine how much, and many billions of dollars are spent on drug research. Just imagine how much the proles, the population, could be helped if somebody would spend just $1 billion on educating them about the low-fat vegan diet with no oils. But it doesn't happen. So no one cares about the proles. It's very important. The average stupid prole thinks that the system's designed to help them. No, the system is designed to exploit you. They look at you as just a farm animal. So you need to understand that. Because once you understand that no one is going to help you, then you've got a chance to study the situation and figure out the safest, best path. Okay. The book describes a company. I think he was in charge of one company, $350 million budget. They couldn't make a single drug that was usable. And here's one of the things he says. One disturbing possibility is that we are approaching the end of pharmaceutical medicine. New drug approvals have been declining for years. One possibility that could open up horizons would be to make better drugs. For making better drugs is if we could figure out how to get small drugs to bind big proteins. In general, small drugs are very, very, very small, and proteins are very, very, very big. And he says this has been called the problem of undruggable proteins. They can't get these little drugs to bind effectively to big proteins, and that's what they think they need to do to get new therapeutic beneficial effects. And drugs, once they go off patent, they don't make much money off them. And so here's a good metaphor. If a protein were a life-size statue, a drug, small molecule, would be the size of an earplug. A typical research result would be a company tries out 530,000 small molecules to get them to bind to a protein, and zero do. Not a single one works. Okay, that's what I meant by the complexity of human biochemistry, metabolic cycles is mind-boggling, and it all works. Whereas you take a genius like this guy, he will dedicate his entire life, and he probably won't be able to come up with one drug that's useful. Okay? That's how difficult it is to work in these systems, okay? And all the food we eat, it's designed to work perfectly in the system. Why wouldn't you want that food? Okay, then he talks about a computer database of small molecules, less than 14 atoms, let's say 13 atoms or fewer, containing just the common molecules in organic chemicals like carbon, nitrogen, oxygen, sulfur, and chloride. And there's over 970 million of them. But all these common diseases, he says, the more we learn, the more challenging these problems appear. For nowhere in sight is there a druggable target that could cure any of these diseases. He's being honest with you. What he's basically saying is, we have experimented with hundreds and hundreds and hundreds and hundreds of millions of drugs, and we can't cure any of these diseases. Yes, he's telling the truth that drugs are basically BS. Okay? Another point he says, for a small molecule to become a drug, it needs to be stable. It can't be just degraded by digestive enzymes in the gut, for example. It has to be non-toxic, not cause tons of side effects. It has to be absorbed through the gut, for example. Goes into the blood, be properly distributed through the body, be easy to manufacture, to formulate, put into pill form, and be stable on the shelf. It has to have adequate half-life in the human body. It has to traverse certain barriers in the human body, like the gut wall, like the cell wall of individual cells, like the blood-brain barrier if it's going to treat the brain, the blood-testicle barrier if it's going to treat the testicles, to bind to a specific receptor on a plasma cell membrane, or to enter the cell and bind to a protein, or to bind to an extracellular protein. That's a lot to ask of any molecule. That's why it's so close to impossible to get a drug that's actually useful. What they usually do is they pay off a scientist to lie about the results, and then they sell the drug as many as they can until people figure out that it's toxic. Like look at Prozac, for example, fluoxetine, the antidepressant. selective serotonin reuptake inhibitor. A little bit about sizes. 1 angstrom is the size of 1 atom. An angstrom is like 10 to the negative 10th millimeter. A nanometer is 10 to the negative 9th. You can remember nano, and starting with N reminds me of 9. Micrometer is about 1 by 10 to the negative 6th. Okay, so 1 angstrom is about the length of an atom. 1.5 angstrom is a carbon-carbon bond. 7 angstroms is the length of a glucose molecule, just some relative sizes. Proteins typically have about 100 amino acids. Peptides usually means less than 50 amino acids. They're typically 2 to 20 amino acids, because you'll hear those phrases used a lot. So anyways, the point I'm showing you this is, you take this Harvard-trained genius chemist, and he devotes his whole life to it, and he'll probably never make a drug that's useful, okay? That's how difficult it is to make it. And then you say to yourself, well, how did God make hundreds of millions of molecules that work perfectly, okay? Because he's God, okay? We can't figure this out. Humans aren't that smart, okay? And this is what he's basically doing. They try millions and millions and millions of molecules, and they throw them into biological systems, and they almost never can make them work, and most of the ones they claim work don't work. And when they come up with a drug that actually works, what it usually means is they just copy something. Like they can make a copy of thyroid hormone, and that'll work because it was already made by God, so to speak. So I just tell you this because the typical person thinks that these chemists and scientists and factories and companies are so effective and smart at what they do, and in reality, they're just struggling to make a profit, throwing a bunch of mud at the wall, hoping some of it'll stick.